Yep, forwards and backwards.
I'll work it out. Hello, everybody. Thanks for listening to me. Well, you haven't listened to me yet. Get so nervous when doing these things. Thankfully, my wife Meredith always gives me the reassuring words of "Don't forget to wash your face, Jeremy," because it takes my mind off that chocolate stone that might be there you're all staring at. Why do I say that? Because the topic that I'm talking about today is the investigation of dieback disease resistance within Banksia coccinea, and it was my wife Meredith Spencer's she's a primary driver for this project. Her, in collaboration with DBCA's Sarah Barrett down in Albany, came up with the idea of undertaking the project, which was gratefully funded through the National Landcare Program, and could not have been undertaken without really valuable assistance from Giles Hardy's team down at CPSM, and of course with assistance from the Banksia farm down in Mount Barker. Now, the wonderful threat abatement program, or threat abatement plan, identified that there is a need to look at the potential resistance in native species, particularly through the screening of priority species, Action 4.4, which I'm sure we're all very familiar with. It also followed on from work that was undertaken in 2013 by Brian Shearer's team, which was focused on the resistance within a species to pathogens, and based primarily on the observation out in the field of long-term, high-impact infested areas where there's been mass carnage, mass collapse of the vegetation communities. However, we still see these long-standing, mature, healthy, highly susceptible species persisting in those environments. And so the question must be asked, are these guys just the lucky survivors? Have they just simply escaped disease, and they've even been given the term disease escapes, or do they potentially have some sort of internal genetic resistance to the disease that has allowed them to persist into the long term? And so the aim of the project was to investigate that exact question: is there natural resistance within Banksia coccinea? And if we can identify that, then potentially it's going to build a foundation for similar work that can be undertaken on threatened and other priority flora species, and provide some practical methods for on-ground management and restoration. I quickly wanted to say that that introduction from Kerry Ann was really quite timely. It came after the very science-based presentation from Giles. We've seen another very science-based presentation, and ours is very much an operational scale practical application that looks at trying to identify something, but relies on so heavily on that science-based management application as well. We are sort of managing our lands by catching up, because we've kind of cocked it up over the last 200 years, and I think we do have an onus on us. Back to the presentation. We first of all went out and collected seed and isolates. We looked at the long infested high impact sites with surviving individuals, and we also took a couple of soil and tissue samples to collect some of the local pathogen that was actually creating the impact that the seeds were collected from. Kevin Collins from the Banksia farm took all of our cones, arranged them into site and parent tree groups, gave them an individual site number, simulated fire, simulated subsequent rainfall events, teased the seeds out, and then propagated the plants, which were then transported up to Murdoch University and placed in the greenhouse for Giles to kill. We, from the propagation, managed to get 25 to 30 plants per parent. That gave us 5 to 6 replicates from each parent to be screened against each of the 4 Phytophthora species with a control group as well. They were all screened with Phytophthora cinnamomi, Nicotiana, multivora, and Pseudocryptogyria. And the PC isolate, as I say, was collected from Gull Rock National Park and was the A1 mating type, which I believe is one of the less common cinnamomi isolates. All the plants were inoculated twice. So the first inoculation on the 1st of April to see what the results were, and then any survivors from the first round were then repotted and re-inoculated on the 28th of August. We had 146 plants approximately from 26 parents per group. 6, occasionally 5 replicates from each parent per treatment. They were all watered for the first few days, and then 3 times a day out thereafter. And the basic trigger to identify the data was focused basically around time to death, defined as whole plant wilting. The inoculation was undertaken simply by inserting a glass tube into the soil medium, allowing that to stay for 10 days so that any potential root damage could heal. Then the colonised millet seed basically placed into a small hole. You can just see in the bottom corner, just below the spoon there, there's the deep hole that the tube made. So the millet seed was just placed into there, and then all of the plants were placed on the bench top in a randomised manner within the greenhouse, and the trial began. So the first symptoms of disease expression were identified on the 8th of April, so 8 days since inoculation, second week after, but only for Cinnamomi, Multivora, and Nicotiana. Symptoms included foliar wilting, discoloration, stem lesions, etc., etc. And a subset of the plants were then plated out for analysis to determine the cause of death, and none of the control plants died. From Cinnamomi, as you'd expect, very aggressive, all plants died except for 5 from 3 parents. And then from the second inoculation, we lost 2 more plants, but I think very encouragingly, we still had survivors from the second round of inoculation from the same 3 parents that survived previously. So these are the same plants that were previously inoculated, have now withstood the pathogen twice. From Nicotiana, was less aggressive, killed individual plants from all but 2 parents and killed all plants from 2 parents. But we had 57 survivors after the first round, so from 146 down to 57. And after the second inoculation, we lost another, 37, 27? My maths has never been good. But we ended up with 20 plants from 13 parents surviving. Multivora, again, less aggressive, killed plants from all parents except 6, but interestingly didn't kill all plants from any parents, with 120 survivors, and after the second inoculation we still had 106 survivors. So from Pseudocryptogyia, that was the one that did not express until later in the program. We only lost 2 plants in the first round of inoculation, 2 from separate parents, and after the second inoculation there was a significant decrease, I guess. We had 137 plants, so not that much impact, only another 8 deaths. In conclusion, it does look from the trials, which are only supported by visual observation, basically, supported by plating the deaths out to confirm that they were actually Phytophthora related. It does look like there is possible resistance to Phytophthora Cinnamomi in those Banksia coccinea. After 2 rounds of inoculation, we only have 3 surviving plants from 3 parents, but they're surviving, and they've survived 2 rounds of inoculation with Phytophthora. So we are starting to be able to say that we're ruling out the chance, the luck factor there, and that there is some evidence that we should be looking more closely at these guys. From Phytophthora multivora, it is becoming more common around the traps. I'm a Dieback disease interpreter. I do see a lot more multivora around the place. And a comment from Giles is that we're seeing huge variability within the different isolates of multivora, with some ranging in their impact to the plants of actually stimulating growth, right through to others that are highly aggressive. And again, my field-based experience would show I've come across sites, particularly on the south coast, where you look at it and you think, my goodness, that's a massive impact. That's a Phytophthora Cinnamomi infestation. You pull out a sample and it comes back multivora, and you sort of scratch your head and go, wow. So had we used a different disease multivora isolate in that trial, we may have seen some greatly different results, given that multivora was one of the less pathogenic species. So from here, the program really needs to sort of grow. My company was able to source the funding, but as a commercial enterprise, we're often not allowed to have that sort of funding. So we really do need to partner with organisations out there. But we really would like to be able to pot or plant the surviving plants out, cross-pollinate them, propagate, and then yet another set of plants to be able to rescreen and reproduce the project with a larger study group. And that's really where it's at the moment, reproducing the project with a larger study group, potentially looking at cloning the either the surviving plants or cloning the progeny from the cross-pollination if we get to that stage. And also screening for other isolates. The Cinnamomai that was screened was the A1 mating type. It would be good to screen the A2 mating type, which I believe is a more commonly recovered isolate from the lab. And also other Phytophthora species that are becoming more prevalent out in the environment. So Phytophthora crassimura, for example. That is about it for me. Are we staying up here for questions now, Maya? Wherever you are? Yep. All right. Thank you.
So we've got a couple of roaming mics for the benefit of the people dialing in. Hopefully they'll be able to hear the questions if we use these. So I'll invite Louise back up, and
Giles is running away.
Giles is running away. And Giles, in case anybody had any more questions for Giles. And yeah, I guess you could just stick your hand up and we'll bring David and I will bring microphones around the room. Any questions?
Brilliant.
Thanks, Jeremy. It's a great presentation. Terrifying to see plants dying in 2 weeks. But anyway, do you have any viewpoints on genetic engineering and being able to think about particular genes that have resistance, and then looking at creating rather than cloning, but using those sort of techniques to create dieback resistant plants to be reproduced?
Oh, look, Justin, the point that I guess bugger off. The point that I guess I make at the beginning is that the point I make at the beginning is that I'm a bushman, really. I'm a field ecologist, I would like to say. We need to undertake these sort of research programs collaboratively. That's much more a question for the scientists, the lab-based scientists, I guess, as far as genetic engineering. The process, the idea, the concept of being able to find a resistant plant and clone it seems fantastic. I'll pass and direct that to cleverer people than I. Anyone else? Or can we go and sit down?
Hi, Jeremy.
Oh, hello. Could you just speak up? Not morphologically, no. I mean, again, I see them out in the field. Basically, the resistant plants are still alive. The non-resistant plants are generally dead in the infested area, or when you move into the uninfested areas, it's very difficult to tell which one's going to survive. So morphologically, no. As far as cell structures and that sort of thing would be where those differences would occur, and that would again be something that needs to be looked at more closely in the lab, and nothing those sort of cell biology sort of levels.
And what about the distribution? Were they located like in the central part of the population or at the edges?
Well, no, see, I mean, the Banksia coccinea communities are almost as densely packed as we're all sat around our chairs here at the moment. They're almost, the community that we would have been sampling and had been healthy would have been so thick you couldn't walk through it. And so once the disease moves through, you're literally left with, say, one person standing in the middle of this room, everyone else is dead. That's the sort of the pattern. Where they're located, no, there's no real pattern as to where they occur in the community or anything like that. No, not really. Very, very difficult to tell. From the field observations, it is very much chance. And what the evidence that I believe we came out with was a lot of the plants that were resisting the pathogen and still living in the field, probably 30, 40 years after the disease front had moved through, didn't really demonstrate any traits of genetic resistance within it themselves. So, the disease moves around through root-to-root contact, from subsurface contact as well as through the soil. And without excavating that individual's roots and working out what's going on with subsurface drainage, subsurface water, it's very difficult to tell what it was, but not all of them were genetically resistant.
One further question, Jeremy. Did you monitor soil temperature in your data experiment? And the reason for asking this is that many years ago, it was found that Banksia grandis seedlings wouldn't be infected or wouldn't show symptoms of infection if the soil temperature was less than 15 degrees. And I've tried inoculating Banksia grandis fruit in the field in winter, wound inoculating them, and found that even though the inoculant applied, the roots didn't get infected. So there may be temperature effect in there.
Definitely. Again, field-based experience, temperature is critical for Phytophthora. We used to call it a fungus back in the old days because it needs it to be warm and moist, basically, and it is most active in the warm conditions. Did we do temperature of soil monitoring, Giles, in the lab?
No. But it was done over summer, so people were tidy at the time.
We got numbers from some other plants growing in the same glass house.
But definitely, probably something to add. I mean, if we get the opportunity to do it again, that is, definitely another recommendation, potentially to run it over a winter series as well. But I mean, would the soil temperature have any? I mean, testing it in colder temperatures, would that have any? I guess it would benefit in determining and looking at resistance, genetic resistance. I'm not sure, but no, Brett.
Jerry, just so your work, obviously you've given an example, you've looked at Banksia species, but just in terms of your experience in the field, I mean, sorry, maybe this plays online, so just in your experience in the field, how common in terms of other species are we seeing, single survivors or individual survivors? So, what is the potential of, I suppose, small resistance in other species, just in what you've seen in the field?
Look, I think we as interpreters, I'm looking at other disease interpreters in the room, but I think we would all put our hand up and say that we have a list of susceptible species that we use as disease indicator species, your Banksias, your Xanthorrheas, Phytophthora, and the list goes on. And we all have our own individual rating system as how susceptible they are to the disease, kind of developed because on our expectation of how many we are going to see. Behind that disease front, I'm stepping back into the well-infested area here, we're always going to be seeing disease escapes, we term them. It's rarely you see everything, as there's always disease escapes behind magnet horns. There's very regularly disease escapes behind the disease front of a range of species, and that's the primary source of the question. Now, are they lucky buggers, or do they have some sort of genetic resistance in there that's protecting them, and we can expose that?
Hi, Jeremy. Just wondering, you've got 3 very precious little survivors there. Have you been able to re-isolate the Phytophthora from them? Can you confirm that the Phytophthora has actually gone and infected them? And have you had a look at the roots?
Giles is giving me a thumbs up. You beauty, Giles. What? Can you elaborate?
Cinnamomum is still present in these pots. Nice.
And has it had an impact on the roots or the amount of vegetation?
So we can isolate cinnamomum from those pots that healthy plants in, and we want to minimise damage. But you pull the plant out of the pot, there's no real root damage compared to where you've got plants that are cinnamomum. So the roots are reasonably healthy.
So what do you do?
Stop killing Banksias.
And that's why we need to take those 3 and the survivors from the other tests, repop them, cross-pollinate them, and rerun the trial again, because we don't know what's going on.
We've got a couple of minutes left. Is there anyone who has a question for Louise? Yeah.
Louise, I was wondering how other stresses like drought might affect the reflections on leaves.
Yes, definitely we've done trials with that as well. I just didn't present results now, but the one reflectance plot where we had canker obviously affects the stem, it goes into the stem, so it affects the water flow into the stem. So my my suggestion on that is that we actually saw effect of water, the canker affect the water flow, so that's what we saw there actually, and not as such a canker, but yeah. So now we've done trials like that as well, and it depends. There's a big seasonality affecting this as well. So whether the plants are growing well and in summer, then you will see differences in the water content as well as when it is winter time. But yes, it's very effective in that as well.
And we've just got one question from the virtual audience for Jeremy. They wanted to know if you see any other species surviving and what the most common species you see surviving when you're out doing the internet work are.
It varies from site to site because different species have different environments. If you imagine in any one patch of bush, you've got different soil types, you've got different hydrology, you've got different vegetation communities. I would say that the least of the reliable indicators would probably be Xanthorrhoeas, platyphyllas, and on the south coast and Preciones up here. But they will go out just as reliable if they're going on. They're one of my favourite indicator species, most reliable indicator species, but they will also have a large number of plants not on.
Perfect.
Question.
And one last question, and then we're on to the next presentation.
Just for Louise there, with your spectral measurements and whatnot, are you finding differences over different types of environments and soils, and how does the light cloud affect your measurements?
That's a very important question. What we've done in our trials, we actually didn't do all the settings in one go. We did them over 2 days to make sure that it's the same amount of sunlight, because I would definitely have an effect. Whether it's cloudy, whether it's a really hot day, because the solar radiance is very intense here. We haven't done any trials in terms of different soil types or environments, but obviously once we test out the dam reservoir marry or whatever species we want to do, we need that would be the next step.
Can I make a comment?
Yes.
You actually do see differences in spectral signatures on Mary from different provinces. We've got some garden trials down on the south coast with 250, 280 families from 18 provinces, and we do get different spectral signatures across that site in different provinces. So there's quite a lot more work to be done.
Yeah, I agree with
So a lot of things affect, a lot of things, soil type will affect, I think.
Yes, I agree with Giles was saying, is that we just need to do more work because we need to select the great planters and we need to know what it does. But yes, definitely.
Okay.
I think there's one for Jeremy.
Anything? Just go first, quick.
A question for Louise. Myrtle rust, has it been used overseas to look at myrtle rust?
Yes, yes. Actually, there's a very neat study now. It was done by Rennie Hine. He's done his PhD on this. We actually developed the myrtle rust index on the lemon myrtle tree, and his work is actually being registered as open source. So we can actually go and we can download the script, and we can actually try to. Anyone can try and develop their own index on this. I know that Angus Canary over East, he's doing lots of work on this as well now. I watched the presentation from me, I think last week, the week before for National Science Week. So yes, he's definitely doing work on this line as well. I think this is a very important issue because once we don't want myrtle rust yet. So I think it would be good if we can get going with that before it arrives here.
Perfect. Thank you, Jeremy. Thank you, Louise. Thank you, Giles, on behalf of Trina for delivering those talks for us. And we're going to go all the way to Oregon and hear from Sarah Navarro from